The COVID-19 pandemic has paralleled the Great Spanish Flu pandemic of 1918 to 1919 in the United States. Previous historical accounts have strongly suggested a post-viral syndrome. Currently, a post-COVID-19 viral syndrome is unquestionable, which shares many of the characteristics of myalgic encephalomyelitis, or chronic fatigue syndrome that is present globally. The original term for this post-acute sequela of SARS-CoV-2 was termed long haulers by those who were affected with this syndrome, and it is now termed long COVID or PASC. International researchers and clinicians are desperately trying to better understand the pathobiological mechanisms possibly involved in this syndrome. Fortunately, large numbers of individuals have now recovered from the acute coronavirus disease 19 pandemic caused by the severe acute respiratory syndrome coronavirus 2. Unfortunately, there are growing numbers and concerns regarding a subset of these recovered individuals who have varied, persistent, and reoccurring symptoms relating to this pandemic that have termed themselves as long haulers, while the medical community has now termed this condition as long COVID, or post-acute sequela of SARS-CoV-2. It is important to note at this time that there is still not a standardized definition of LC or PASC. These individuals represent a failure to return to a baseline state of health after recovering from acute COVID-19, and encompass various physical, neurologic, and neuropsychiatric symptoms lasting from weeks to months following the acute infection of COVID-19. Typical symptoms of LC or PASC include fatigue, shortness of breath, chest pain, brain fog, sleep disorders, fevers, gastrointestinal symptoms, anxiety and depression that can persist from weeks to months and can range from mild to incapacitating. Because SARS-CoV-2 binds to the cellular angiotensin-converting enzyme 2 receptor that is present in multiple organ systems, LC or PASC has a multifactorial pathogenesis, which incorporates chronic inflammation both in the peripheral organ systems as well as in the CNS. Neuronal system dysfunction, specifically including the activation of neuroglial cells, is associated with neuroinflammation and impaired cognition, and the activation or dysfunction of brain endothelial cells are associated with subsequent inflammatory thromboembolism. Thus, the activation of neuroglial microglia and astrocytes are thought to be key pathogenetic mechanisms in LC or PASC. The PCCs are thought to be elevated in LC or PASC individuals, and they may occur in a continuous process or in recurrent wave-like processes that affect the brain. Microglia cells possess a unique quality to undergo rapid changes in their diversified spectrum of varying ultrastructural phenotypes. They may exist in a ramified state, a polarized activated amoeboid state, and an aged senescent morphological state. Ramified microglia cells serve continuously as the housekeepers and cleaners of normally accumulating cellular and metabolic debris. They are the innate immune cells of the central nervous system and are in constant surveillance of their surroundings in order to detect any type of damage or injury within the central nervous system, including the neurovascular unit and the blood-brain barrier. The endothelial glycocalyx is vasculoprotective and acts as the first barrier of the tripartite BBB. The ECGCX serves as a protective coating, prevents the direct contact of the circulating blood and its constituents with the plasma membrane of the brain endothelial cells. This gel-like mesh network is responsible for multiple other protective functions in the vasculature, including the promotion of vascular integrity and homeostasis, signaling blood and vessel wall capillary interactions, providing an anti-inflammatory, antithrombotic, and anticontractile surface layer, limiting access of certain molecules based on size, steric hindrance, and especially electrostatic charge. Since the glycocalyx has a strong negative net charge and is capable of repelling highly negative charged molecules and mechanotransduction of fluid shear stress, and subsequent maintenance of bioavailable nitric oxide to the vessel wall. Importantly, the ECGCX is capable of modulating inflammatory processes via its prevention of leukocyte and platelet adhesion. It is also important to note that the ECGCX and epithelial glycocalyx are specialized extracellular matrices containing numerous proteoglycans, such as chondroitin sulfate proteoglycans, tenascinar, hyaluronan, and link proteins termed the perineuronal net. These PNNs are comprised of numerous compressed scaffolding of synapses, and are certainly very vulnerable to the detrimental effects from pro-inflammatory CNSCC that are associated with increased oxidative stress provided by the pro-inflammatory. PNNs have multiple roles, but the most critical are the regulation of synaptic plasticity via stabilizing synapses and, further, provide protection of neurons and synaptic connections against damaging stressors. Homocysteine is a non-essential sulfur-containing amino acid and an intermediary metabolic product derived from the demethylated essential amino acid methionine. Plasma concentrations of homocysteine are inversely related to plasma concentrations of folate, vitamin B12, and vitamin B6, as well as to the intake of these vitamins. Hyperhomocyteinemia has vasculotoxic effects, as well as neurotoxic effects that are associated with neuroinflammation, neurodegeneration, pro-oxidation, as well as protherogenic/prothrombotic effects. Elevations of HCY or hyperhomocysteinemia plays an important role in the causation of oxidative stress with excess formation of reactive oxygen-nitrogen species in the endothelium and in the central nervous system, due to autoxidation of HCY, formation of HCY mixed disulfides, and interactions of HCY thiolactones and protein homocysteinylation. Micronutrients consist primarily of vitamins and minerals and play an important role in CNS homeostasis. Micronutrients are an essential component of several general cellular functions in addition to proper functioning of neurologic activity, such as the synthesis of certain neurotransmitters including dopamine, serotonin, myelin formation, synaptic plasticity, and energy production, which is decreased in many neurodegenerative diseases. When there is insufficient uptake or depletion of micronutrients, the brain cannot continue to function normally, and the result will be dysfunction or disease, that is a loss of homeostasis, such as in the dysfunction of FOCM, which occurs in both acute COVID-19 and post-COVID-19 LC or PASC. Furthermore, as SARS-CoV-2 infects host cells, it hijacks the normal 1-carbon cellular metabolic machinery. This places significant demands on the host cell methyl groups in order to replicate its ribonucleic acid and provide RNA capping to produce large numbers of progeny virions. As one begins to better understand the impaired 1-carbon metabolism hypothesis in COVID-19, it becomes apparent that supplementation to patients suffering from LC or PASC with vitamin B12 and folate might have positive responses due to the impairments caused by this syndrome. For example, Regland et al. demonstrated that individuals suffering from myalgic encephalomyelitis and fibromyalgia, that is chronic fatigue syndrome with symptoms similar to LC or PASC, had a positive response of their symptoms when treated with injectable B12 and oral folic acid. Systemic PCC and CNSCC in COVID-19 are known to induce endothelial cell activation and an inflammatory-related immunothrombotic or thromboembolic state within the vasculature. This immunothrombotic state ultimately causes macro-microthrombotic complications in multiple organ systems including the central nervous system, which may relate to either macrovascular or microvascular thrombi or clots. Microvascular thrombi may also be associated with microbleeds, especially in the white matter regions. These thrombi or microbleeds associated with cerebral small vessel disease will result in ischemic injury and ischemia/ischemia reperfusion injury due to decreased cerebral blood flow and impaired metabolic delivery and metabolic byproduct uptake that may induce even further neuroinflammation due to increased CNSCC. This increase in CNSCC will develop in a response to injury wound healing mechanism due to activated neuroglia, which secrete CNSCC and reactive oxygen-nitrogen species, resulting in ischemia-related regional injuries, due to decreased cerebral blood flow to evoke even further neurodegeneration and/or impaired cognition. Our global societies have been facing the COVID-19 pandemic now for 17 months, and the concerns regarding the disability of individuals with LC or PASC, and their increasing numbers are of great concern. Researchers and clinicians are trying to better understand the multiple pathobiologies of LC or PASC in an attempt to be better informed as to how to treat these individuals. Indeed, repurposing or repositioning of existing medications is not a new concept, and one possible treatment is to suppress the chronic peripheral PCC and CNSCC neuroglia activation, neuroinflammation, and impaired cognition that may be central to the syndrome of LC or PASC. One example is to utilize anti-inflammatory modalities along with the supplementation of micronutrients, such as vitamin B12 and B9, folic acid, to assist in 1-carbon metabolism to treat individuals affected with this syndrome. The author has had his own personal experiences regarding a post-viral syndrome during the recovery from herpes simplex encephalitis and personal experiences in treating those patients with a significant post-viral syndrome due to human cytomegalic virus with upper respiratory infection similar to influenza, in which affected patients were fatigued and suffered from impaired cognition, who responded positively with injectable methylprednisolone and B12 and oral folic acid. The concept for repurposing methylprednisolone, vitamin B12, and folic acid began with 2 personal experiences by the author. The first experience was my own rapid recovery with symptoms of post-viral chronic fatigue syndrome similar to long COVID or post-acute sequela of SARS-CoV-2 syndrome. Upon surviving my own experience with herpes simplex type I encephalitis in 1997. The second experience by the author involved a small isolated community outbreak of the human cytomegalic virus. In 1999, author experienced a small community outbreak of the human cytomegalic virus with a total of 6 patients. This small community outbreak of 6 patients with HCMV and post-viral syndrome, in addition to their similar history and physical findings, suggest to the author that these similar treatment modalities might be successful in the treatment of LC or PASC, and at the very least should be given serious consideration. Of course, time heals all wounds. However, the LC or PASC certainly seems to have a slow, long and winding road to recovery. Probably one of best known of all viral infections that has a prolonged post-viral syndrome of fatigue and myalgias is Epstein-Barr virus infection. In these cases, the author has repeatedly witnessed the positive effects of anti-inflammatory methylprednisolone acetate and vitamin B12 injections as previously described. It is important to note that the use of corticosteroids have not been confirmed by any randomized clinical trials to date in regards to the prolonged nature of the post-viral fatigue, and in regards to their usage one should weigh the benefits versus the risk in each individual case presentation. Importantly, many of these patients will require a program of progressive physical therapy to help regain their pre-COVID physical activity and endurance. Professional healthcare teams who particularly have an older senior citizen clientele should consider reading a paper by a noted gerontologist